Hello and welcome to Pharmacogenomics. In this video, we are going to have a look on some important information on pharmacogenomics. This is one of the topics that are covered in many entrance examinations. Now let's look into some important definitions before going to the main part. What is precision medicine? It is an approach to disease treatment and prevention that takes in consideration genes, environment, and lifestyle. Pharmacogenomics. It is a component of precision medicine. Study relation or variation in multiple genes and variability of drug response. Pharmacogenetics. It is a study relation or variation in single gene and variability of drug response. Here pharmacogenetics is variation in single gene, whereas pharmacogenomics is variation in multiple genes. Now let's look at the components. Nucleotide. It is a basic structural unit of DNA and RNA. Gene is a stretch of nucleotides that codes for a single protein. DNA is genetic material that is the main component of chromosome and is double helix. Traits in pharmacogenetics are classified as genotype, set of unique genes that determine specific trait in a person, such as creativity. Phenotype, observable trait of genotype, gene responsible for blue eye color. What is the difference? Genotype is a unique trait, whereas phenotype is observable trait. Other important terms you need to know are polymorphism. Genetic variation is common 1% or more, and genetic variation is inherited mutation. Genetic variation is rare, less than 1%. Single nucleotide polymorphism, SNP, is change in a single nucleotide in a genetic sequence. Homozygous means two alleles are identical. Heterozygous means two alleles are different. Now let's look at some drugs and variations in response to drugs in individuals with genetic variations. First, let's look for HLA-B gene. Human leukocyte antigen B is a normal gene. It is essential for immune system. Any variations in HLA-B gene leads to variations in drug response. Abacavir, HLA-B, 5701, if you have HLA-B, 5701, then there is increased risk of hypersensitivity. That is, in individuals having HLA-B 5701 gene, abacavir shows hypersensitivity as side effects. Again, same is for allopurinol. Allopurinol HLA-B 5801 gene shows hypersensitivity. Both of these drugs shows hypersensitivity reactions. Only difference is between the numbers 5701 and 5801. Carbamazepine and oxcarbazepine. HLA-B 1502, which is an abnormal form of HLA-B gene. Then it leads to severe dermatologic reactions such as Stevens-Johnson syndrome, hence it is not prescribed in individuals showing HLA-B 1502 gene. Population at risk. South Asian usually express this gene and are at high risk. Phenytoin and phosphonotoin gene HLA-B 1502 also shows hypersensitivity. These drugs were showing response to specific HLA-B gene. Both have the same gene 1502. Now let's look into other class of gene, such as CYP gene. Clopidogrel, CYP2C19-2 and CYP2C19-3. Clopidogrel is a prodrug that needs CYP2C19-1 for activation. If CYP2C19-2 and 3 are present instead of CYP2C19-1, then clopidogrel will not be converted into the active form. Hence, there is increased risk of cardiovascular events due to no antiplatelet effect of clopidogrel. Voriconazole, CYP2C19. Patients with this gene mutation have 2-folds to 4-folds higher voriconazole exposure. However, no dose adjustment required or no genetic recommendations. These drugs were specific to CYP2C19. Warfarin. 3 genes CYP2C92, CYP2C93, VCORC1. Increase CYP2C92, 3 activity, then reduce warfarin metabolism. In turn leads to bleeding. Increase VCORC1 activity or expression leads to increased warfarin sensitivity, which in turn leads to increased bleeding. For this, lower doses should be started. Celecoxib CYP2C9. If a person has increased CYP2C9-3 expression, then there is reduced celecoxib clearance. For this, it is recommended to use with caution and at lower doses. These drugs are specific to CYP2C9 gene. Coding codes for CYP2D6 enzyme. Increase CYP2D6, then increase metabolism of codeine to morphine. And if there is decrease in CYP2D6, then metabolism of codeine to morphine is reduced. Hence, low or no effect of codeine since it is not converted to morphine. However, morphine high dose cause side effects such as respiratory center depression. Other drugs such as risperidone, atomoxetine, fluoxetine and tamoxifen. CYP2D6, individuals with increased CYP2D6 are fast metabolizers, have high exposure to these drugs. While those with reduced CYP2D6 are poor metabolizers of these drugs. These drugs were specific to CYP2D6 gene. Other class of gene include HER2. Drugs such as trastuzumab, pertuzumab, lapatinib and emtansine. HER2. If an individual shows HER2 positive, then take these drugs. If HER2 negative, then do not take these drugs as it shows different or no response. Azathioprine and 6-mercaptopurine require TPMT enzyme. Individuals with reduced TPMT, there is increased risk of myelosuppression. Drug should be not given or given at low dose. Capecitabine and 5-fluorouracil. DPD gene. Reduced DPD leads to severe toxicity of these two drugs, as well as diarrhea, neutropenia, and neurotoxicity. For primaquine and rasburicase G6PD gene. In individuals with reduced G6PD, there is risk of hemolytic anemia. At-risk population are African and Mediterranean. For cetuximab and panitumumab. KRAS mutation. If individual shows KRAS positive, then no response to these drugs. So do not use in such individuals as it shows no response. KRAS negative, then these drugs shows response. It can be used. KRAS is G protein in the EGFR pathway. EGFR is anti-epidermal growth factor receptor. Again, cetuximab and panitumumab inhibit EGFR anti-epidermal growth factor receptor, blocking growth factor binding, receptor activation and subsequent signal transduction events leading to cell proliferation. Imatinib CD117. Imatinib is indicated for patients with CD117 mutation. That is, it is given in patients showing CD117 mutations. Maraviroc CCR5 gene. Maraviroc is a CCR5 receptor antagonist. It acts as an antagonist at CCR5 receptor, which block R5 tropic HIV entry into CD4 cells. Irinotecan UGT1A128. Increased UGT1A128 activity or expression leads to neutropenia, and homozygous individuals are at higher risk. Lenalidomide chromosome 5q deletion. Chromosome 5q deletion leads to myelodysplastic syndrome and hematologic toxicity. Now the easiest way to remember is HER2. Use the drug if individual is positive for HER2 and avoid if negative. TPMT. Reduced TPMT, then there is increased risk of myelosuppression. DPD. In individuals with reduced DPD. Indicates severe toxicity, DNN, that is diarrhea, neutropenia, and neurotoxicity. G6PD. Individuals with reduced G6PD activity. Then there is risk of hemolytic anemia. HLA-B. Avoid the drug if individual has positive HLA-B gene. KRAS. Avoid the drug if individual has positive KRAS. You can remember as avoid if positive for HLA-B and KRAS. Avoid if negative for HER2. These are few basic important notes you need to know while you are appearing for any exam, especially licensing exams. Analyze yourself by taking the Q and A session in the next video. Do visit my website www.pharmadip.com. Like, share, and if you haven't subscribed, don't forget to subscribe and remain updated on what's coming next.